Thanks, Barry. So I've learned a lot today. I have a feeling in 40 minutes I'm going to learn a little bit more. And what I'll try to do during the talk is to contrast two hormone-sensitive systems in the brain. Steroid sex hormones, I'll use estrogens as the example, where the test system is a simple behavior that controls reproduction in laboratory animals. We've got the hormone receptors, the neural circuitry, and transcriptional changes already. And so we feel that insofar as we can do business with histone modifications, that we can read it back all the way to behavior without too much difficulty. But then I'll contrast and oppose that to steroid sex hormones, glucocorticoids, where in this case the test system is restraint stress in laboratory animals, and the readout leads to hypotheses that are interesting, at least to us, and they're going to have to do with a phrase that I'll call genomic stress responses. So with respect to estrogens, the system we set up is brutally simple. If the female laboratory animal has enough estrogens on board, then she'll do this vertebral dorsiflexion, which is essential to reproduction. Without this lordosis behavior, the male cannot fertilize. And using this as a hobby horse, we've been able to figure out how steroid hormones coordinate brain function with the rest of the body to ensure reproduction which is appropriate to the environment. And we understand the mechanisms from receptor chemistry, where the ligand binding pit is measured in angstroms, through the seasonality of reproduction, where the relevant physical variable is light years. And so the whole field can be summarized in 3 steps. The first step would be discovering hormone receptors in the brain. What you have is sagittal sections of a rat brain on the midline and farther lateral. And what we found, in this case with estrogens, is a limbic hypothalamic system of neurons that bind radioactive estrogens. And for this purpose, today's purpose, in the limbic hypothalamic system, which as I said is true among all vertebrates, the only important cell group is marked down here by VM, which is the ventromedial nucleus of the hypothalamus. And the reason that's important for today is that for working out the neural circuit that governs lordosis behavior, which was the first neural circuit for any vertebrate behavior, we had 3 places to start. The first place was these hormone-dependent cells in the ventromedial nucleus of the hypothalamus. The second place was to follow the stimuli in using neuroanatomical and neurophysiological methods. And the third anchor for discovering the circuit was to follow the motor system backwards through the motor neurons, through the reticulospinal neurons. And what we ended up with was a spinal, midbrain spinal circuit, which is controlled by an estrogen-dependent signal coming from the hypothalamus. So it took years to do this, but then we got very, very lucky, because it turned out that the hormone receptors we had discovered are ligand-activated transcription factors. So over the years, we could discover a list of genes that has 2 properties. The first property is that estrogens, having bound to primarily ER alpha, but in some cases ER beta, will elevate transcript levels, except for this one down here, which works by disinhibition. And the second property is that the gene product fosters female reproductive behaviors. And of course, the big question is exactly how do these transcriptional effects occur? How are they regulated? And so I'll use this simple cartoon to tell you the kinds of things we already know. We know that estrogen receptors, if and only if they're liganded by estrogens, will go from the cytoplasm into the cell nucleus. They'll dimerize, and through their DNA binding domains, they'll attach to an estrogen response element. And what I have here is so-called consensus estrogen response element, which is not a palindrome. It has reverse symmetry. It's not a palindrome. And also, substitutions are allowed in this. Some of the bases can be substituted freely. Others have to remain as they are. Glucocorticoid receptors, very similar kind of molecular dynamics, except of course the GRE, glucocorticoid response element, is different from the ERE. And the other thing to mention is that thyroid hormone receptors, where the dominant TR in brain is thyroid hormone receptor alpha 1, as discovered by Bjorn Wenstrom at the Karolinska Institute, has as its favorite response element the 5' end of the ERE, the estrogen response element. So that's something I'll bring into play in a few slides. So since we're dealing with a behavior where the effects of estrogen on that behavior absolutely depend upon new RNA synthesis and new protein synthesis, what we're obviously looking for is pro-transcriptional events, or shall I say at least histone modifications which are correlated with increased transcription. So it was very nice that when Zach Weil came to the lab, studying global histone acetylation so that we could do immunocytochemistry, he did see a significant effect of estrogens on H4 acetylation in the ventromedial nucleus of the hypothalamus, which is relevant for lordosis behavior. And we're antibody limited, so I hope you can see the positive cells for PAN4 acetyl. Here you can see the immunocytochemistry for ER alpha, and here you see the merge. And so here, every yellow dot is a cell in the ventromedial nucleus of, this is the bottom of the brain right here, in the ventromedial nucleus of the hypothalamus, in fact, in the ventrolateral corner of the ventromedial nucleus of the hypothalamus, which both has ER alpha and PAN4. So we were encouraged, and we continued with global acetylation measures. Katuna Gagnidze in the lab discovered positive estrogen effects on acetylation, on a combined acetylation and phosphorylation event. Both of these are associated with increased transcription. And then this H4K9 trimethyl, which is a repressive mark, we were very pleased that it went down this amount of time after estrogen treatment. We cannot explain this bar, and we cannot explain that bar. So we couldn't go on doing global acetylation forever. And I forgot to mention that in that laundry list of genes, two of our favorites in terms of robustness of induction and also close association with lordosis behavior are the progesterone receptor and the oxytocin receptor. So those promoters were favorite choices for us to do histone work. So here's the induction of the protein, the progesterone receptor protein, two splice variants, so-called big one, PRB and PRA, and these are relatively large inductions for the nervous system. Most of you know, at least with respect to hormone effects, that inductions in the peripheral tissues are always larger than inductions in the brain, as you would expect from a heavily interconnected, highly regulated organ. So the question is, how are we going to study the progesterone receptor promoter? Obviously, we use CHIP with immunoprecipitation. The first data I'll show is from an acetylated histone. And then, of course, we're choosing places on the progesterone receptor promoter to do PCR. So here are the 4 places that we chose to interrogate with CHIP, and the results as a function of estrogen treatment related to lordosis behavior in the ventromedial nucleus of the hypothalamus, the same cell group as regulates lordosis, depend upon which mark you're looking at and the amount of time after estrogen. So you can see the only significant effect we got here with the H3 acetyl mark was on this promoter segment 3 hours after estrogen. The numerically greatest effect was here farther downstream, but there was too much variability for it to be significant. But then a different mark, a pro-transcriptional H3K4 trimethylation, the best effect was far downstream and at 5 hours after estrogen treatment. Likewise with the oxytocin receptor promoter, here are the 3 places we chose to interrogate, and with the acetyl, H3 acetyl antibody, we got our best effects at 3 hours after estrogen treatment, and our best effect with this pro-transcriptional H3K4 trimethyl, the best effects always at 5 hours, especially farther downstream. So this was much different than that, for example. So when estrogen receptors, or for that matter, other nuclear hormone receptors bind to the DNA, they're always surrounded by other proteins which in the molecular endocrine world are called coactivators. But those coactivators tend to be histone acetyltransferases or pro-transcriptional histone methyltransferases. And we were pleased that we got something more than a 50% increase in messenger RNA for these here and here. And so that tended to support the notion that we really were studying epigenetic events as a function of estrogen treatment, which would lead to increased transcription. Nevertheless, I was looking for my life to get simpler. You noted the complexity of those data, and did we have a unitary explanation of how this behavior occurs out of those data? The answer was no. But Larissa Costa Faustino came to the lab, a Brazilian graduate student, and she wanted to study thyroid hormone relationships in the brain to estrogen-facilitated responses, which of course made our life harder. And the reason I could justify it was that some years before that, we had discovered that hyperthyroid conditions inhibit estrogen-facilitated lordosis behavior. And as I mentioned to you, the 5' end of an ERE, also it's the highest affinity binder for TR-alpha-1 according to Bjorn Venstrom. And so our hypothesis was, our theory was, does competitive TR-ER binding explain the molecular endocrine data, or for that matter, the behavioral data? And some of the data could be predicted and some not. Here at the messenger RNA level, always in VMH for its relevant to the lordosis behavior, yes, we did get a nice induction of progesterone receptor mRNA as expected. And in the hyperthyroid condition, as predicted, we abolished that. And in the hypothyroid condition, we retained it. However, over here on the oxytocin receptor messenger RNA measurements, we got the estrogen induction nicely, and the hyperthyroid condition did abolish the estrogen condition, but the hypothyroid condition did as well. At the CHIP level as well, this slide is set up exactly the same as about 3 slides ago, except now we're looking at TR-ER relationships in the ventromedial hypothalamus on the progesterone receptor promoter. And to give an example of our complexity, yes, we did get a nice increase. We replicated the increase in the H3 acetylated mark in the control condition, and it was indeed abolished in the hyperthyroid condition, but it was also abolished in the hypothyroid condition, and that hypothyroid result was not supposed to happen. Likewise, over here with the H3K4 trimethyl, here's the estrogen induction. We replicate that, and the hyperthyroid condition ruins it, but the hypothyroid condition does as well. So at that point, I felt that we were working very, very hard for results which were not crystal clear. That is, that they didn't explain the behavior in 2 or 3 steps. And I reflected for a few minutes on the difficulties of doing molecular biology in the intact nervous system. The technical issues are that we're always starting out with small amounts of starting material. We have to, in order to have anatomical particularity, and that's even before you take out an individual cell type. We have heterogeneity of starting material. In the TR-ER experiments of Larissa Costa-Faustino, we had to do a frightening amount of normalization. And I don't know about you, but the more you fuss with the numbers, the more nervous you could get. And finally, we're always relying on experts working with other tissues. At Rockefeller, I'm always asking Dave Alice silly questions and begging reagents and everything else. And in molecular neuroendocrinology, we're always reliant on the concepts and the reagents developed by estrogen experts studying breast cancer cells and androgen experts studying prostate cancer cells. So we always feel as though we're on thin ice. Conceptually as well, we tend to study in any given experiment with just one level of regulation. Whereas when we're talking about transcription factors and the corresponding epigenetic changes, we have transcription, we have translation, which is regulated. We have the exact position of the protein in the cell nucleus. It's not just a solution. So if you look at the work of Gordon Hager at NIH, for example, in the GR field, glucocorticoid receptor field, you'll see that the exquisite control over protein condition during transcriptional events. And of course, then you have the availability of the binding surfaces. A couple of angstroms can make all the difference. The order of events, acetylation, methylation, phosphorylation, and their interactions. And finally, even in the hormone receptor field, we're lacking some of the specific inhibitors of co-activator receptor interactions. Tom used the phrase epigenetic pharmacology, and in our field, for the most part, we don't have the tools yet. Nevertheless, when we came back to these ventromedial hypothalamic neurons with a pharmacologic approach, we were able to get a very clear, behaviorally important result that made sense. We were saying that increased acetylation should foster lordosis behavior. Therefore, if Zachary Weil would set up animals where into the hypothalamus, we set up animals who were overreactomized, so they had no native estrogen of their own, and we gave them estrogen levels which would prime their lordosis behavior, but it wasn't a high enough dose for lordosis behavior to actually occur. So we fixed it so that they were close to being behaviorally competent, but they weren't all the way there. And then we infused into the ventromedial hypothalamus artificial cerebrospinal fluid, which is a frequent control that you use in the brain. And we got 0 lordosis. That's what we wanted. We wanted a flat baseline, a clean baseline. And then when we injected the histone deacetylase inhibitor, trichostatic acid, we got lordosis behavior in almost half the animals. Furthermore, when female laboratory animals do not want to be mounted by males, they usually kick the males in the rib, or they turn around and do a boxing posture. And we call those rejections, familiar to all of us. And the percentage of rejections goes down. So when we came back with a pharmacologic approach, we were able to do business in a much easier fashion. So summarizing the first half of the talk, we have these hormone receptors, these estrogen receptors in the brain, and it's the expression of the gene ER alpha, which is responsible for the estrogen binding and transcriptional effects in the ventromedial hypothalamus. So we focus on those neurons in the ventrolateral corner of the ventromedial nucleus of the hypothalamus, which express ER alpha, and whose effects are read out through a determined neural circuit, which we worked out over a period of years, in order to regulate a specific behavior, lordosis behavior. We have several estrogen-regulated transcriptional events, some easier to study than others, that foster lordosis behavior. And so far, we have two pro-transcriptional histone N-terminus modifications, more to come. The next question is, over the coming years, to what extent do these histone modifications form a coherent story in order to explain the estrogen effects on lordosis? And that's an open question. I said at the beginning that I would contrast the two systems, the sex hormone-dependent systems and the stress systems. And so my way of saying it is that with respect to sex behavior and estrogen effects on brain, you have preparations for reproduction spread throughout several kinds of cells in the body and over a long period of time. And they all converge in order to regulate a single event, which happens within a couple of hours as the lights turn to dark on the afternoon of proestrus. Out of a 4-day estrus cycle, all the action happens at the end of the afternoon and just after the lights go out, which is ovulated correlated with sex behavior. It guarantees that when the female goes out of the burrow in order to mate, it's likely to be a productive mating because she just ovulated. In contrast, with respect to stress, a single stressful event can have effects which are spread throughout the body and which can act a distressingly long period of time. So not only can we contrast the physiologies of these two systems, but also stress blocks female reproductive processes. It's a famous fact that stress operating through corticosterone or cortisol can inhibit the ovulatory luteinizing hormone surge coming from the pituitary gland, and it also inhibits lordosis behavior. And we were wondering how it inhibits lordosis behavior, or for that matter, female reproductive physiology in general. And Bruce McEwen's favorite phrase is a life history of emergencies. In other words, in the clinical condition, you have reproductive processes that are interfered with by a lot of psychosocial events. Getting more concrete with respect to animal behaviors, we know that isolation stress reduces oxytocin receptor messenger RNA. We know that there's glucocorticoid response elements on the progesterone receptor and the oxytocin receptor promoters. And what we don't know yet and we'd like to discover is whether there are epigenetic changes from stress through the PR promoter or through the oxytocin receptor promoter. So having contrasted them, I'd now like to remind you that when cortisol in the human or corticosterone in a laboratory animal reach the brain, they're not only exerting negative feedback through the hypothalamus, but they're also regulating a bunch of behaviors. So what corticosterone does is to go to the paraventricular nucleus of the hypothalamus and turn off a synthesis and release of corticotropic releasing hormone or vasopressin, thus to reduce the amount of ACTH coming from the pituitary, the anterior pituitary, thus to reduce the synthesis of corticosterone. So that's a negative feedback. But as well, corticosterone is operating in the hippocampus and in other parts of the limbic system and the cortex in order to regulate a variety of important behaviors. And Bruce McEwen discovered binding of corticosterone in the hippocampus. Here we have the dentate gyrus, the granule cells of the dentate gyrus, and here we have Ammons horn, the pyramidal cells of Ammons horn. And he emphasized glucocorticoid receptors mediating that binding. But then Ron de Kloot came along, he's at Leiden in the Netherlands, and he discovered that some of the binding is also due to mineralocorticoid receptors. So what you have is a two-receptor system, a high-affinity, low-capacity system binding corticosterone, the mineralocorticoid system, and also GR, the glucocorticoid receptor, with lower affinity and higher capacity. And the impact of this in the hippocampus can be severe. What you have here is a representative pyramidal cell from the hippocampus of a control animal, and here an animal who's been subjected to a chronic psychosocial stress. He's a subordinate male in a severely aggressive situation. And you see the denuded dendritic tree, severely denuded dendritic tree from a paper about 20 years ago. So why do we think of this in an epigenetic framework? And the reason is that for stress-related neuropsychiatric diseases, it's really been difficult to demonstrate very large contributions of individual genes to the susceptibility to those stress-related diseases. And the thing I know most about is autism, where 5, 10, 15 years ago, geneticists went at it in order to discover the autism gene. But now if you summarize the work of the wonderful Evan Eichler, who's at Washington University, and he would say, how many genes could be associated with autism in little boys? And the answer would be about 800, including a lot of de novo mutations. So it's really difficult. And so you could say that that's, I don't think that Tom Inso would agree with me, but you could say that that's a defeat. At least I don't want to do that. So what I'd rather do is look above and around the genes and try to use epigenetics in order to get simpler results and simpler concepts that contribute to what we could eventually call epigenetic pharmacology. So the leader of this work is Richard Hunter. Richard worked with Bruce and with me, and he's now an assistant professor at the University of Massachusetts. And he worked with 3 different moieties. I've already talked about the protranscriptional H3K4 trimethyl. And for the rest of this talk, to stay within time, I'll concentrate on the repressive effects of H3K9 trimethyl. And our model is acute restraint stress over 30 minutes, followed by sacrifice of 1 hour recovery. And then the other thing we did was chronic stress, which is 2 hours of immobilization every day for 21 days. And since this is what they call a homotypic stressor, the animals show an adaptation of the hypothalamic pituitary adrenal axis and also behavioral adaptation. That's important to know. So the result upon which we've hung a lot of studies is this one. This was published about 4 or 5 years ago. And it is that acute stress increases trimethylated H3K9 binding in the hippocampus. Here's a half of a hippocampus taken from a control animal. Here is the dentate gyrus. Here's Ammon's horn. And you see the relative lack of immunoreactivity using this antibody. Whereas in the acutely restraint-stressed animal, we have great density, optical density, in the dentate gyrus and also increased optical density in the pyramidal cells. The quantification is here. For the dentate gyrus, a significant increase after acute restraint stress. And here with the monomethylation, well, those are different enzymes that do the monomethylation and the dimethylation. And so I don't know the reason for this going down, but I would like to consider that this could be a source-sink relationship, this being the source and this being the sink, but we haven't proven that. As I said, the system adapts, and with respect to H3K9 trimethylation as well, in the dentate gyrus, here you have the control, here you have the acute stress result, more than a doubling, and with 7-day or 21 days of stress, the system adapts. Which enzymes are responsible? Well, for the trimethylation, you're talking about Suvar39H1 and Suvar39H2, and what you have in the upper left of this slide is a low-resolution film autoradiogram measuring the messenger RNA for Suvar39H2. This half of the slide was taken from a control animal, and this half of the slide was taken from an acutely restraint-stressed animal. Here's the dentate gyrus, and here is the pyramidal cells. The quantification for the dentate gyrus is here. The quantification for the increase in the pyramidal cells is here. And these results could be explained, but we don't know that they are explained by the binding, as determined by ChIP-seq, by the binding of glucocorticoid receptor coupled with, liganded with corticosterone to the region of chromosome 17 in which the Suvar39 H2 gene is located. But we haven't proven that this is the cause of that. So what Richard wants to put forward, based on these data and the literature that he knows about, is that what we're talking about is a genomic stress response. And insofar as the destabilizing elements that we would like to keep in place are transposons, we fully recognize that the thinking that Richard and our team is doing is different than, probably different than what Barbara McClintock meant. It's certainly different from what the first cloner of a transposon meant, Nina Fedorov. And our emphasis is somewhat different than Rusty Gage's in his 2013 Journal of Neuroscience paper, in which he anticipates, if I read it right, that the presence of transposons could lead to capacities for neuroplasticity which otherwise wouldn't be there. Is that about right? Or did I mangle that? Yeah, okay. So let's see where Richard's thinking takes us. The right side of this slide is fact, and the left side of the slide goes towards supposition. The right side of the slide reminds you that as we go from yeast through flies to mammals, the percentage of the genome which is devoted to coding regions goes steadily down to, I think, less than 5%. We know that H3K9 trimethyl is typically associated with heterochromatin. We think that heterochromatin in vertebrates is thought to help maintain genomic stability. And Richard's hypothesis is that heterochromatin formation and maintenance could be part of what he'd like to call a genomic stress response. And I'm off my patch when I try to summarize this genomics paper from 2007, but classifying the different types of retrotransposons. We have the long interspersed nuclear elements, the lines and the signs. We have the long terminal repeats. We have the DNA transposons, rolling circle transposons, self-synthesizing transposons. And sometimes a neuroscientist can understand some of this. For instance, oxytocin and vasopressin are two very important neuropeptides for social behavior and other forms of emotional behavior. They're manufactured on the same chromosome, and there's a long interspersed nuclear element aligned between them. And so that almost certainly means something important. It's something we can relate to. But the rest of this is something that I just don't understand. But I do understand that Helen Rowe and her team in cell culture some years ago knocked out a gene called CAP1. And the result of that CAP1 was to abolish H3K9 trimethyl. And she thinks, and we think as well, that as a result of losing H3K9 trimethyl, that these retrotransposons, the IAP and the line 1, were greatly increased over the 3T3 cell controls. So thus encouraged, we wanted to go ahead and try to, quotes, map a genomic stress response in the hippocampus of the mammal. And so what we did, of course, was ChIP-seq. And what we did was we compared the normal genomic DNA with control unstressed animals, all of this in the hippocampus, and then with acutely stressed animals. The result yielded 30 million 75 base pair reads per group, and you can see the increased binding of H3K9 trimethyl following stress. So then what Richard did was he aligned the positive reads with rodent repeats and the endogenous retrovirus database, and he got more than 20. I hope you can read this, but I'm not going to obviously go down it. But he got more than 20 greatly increased retrotransposons, some of them increased by tenfold or more. Looking at the raw data with higher resolution, on the left side, on chromosome 12, on the left side we have a segment which is in the region where there's a whole lot of genes transcribed. And you see that the stress H3K9 trimethyl reading compared to the control is not very different. But when we go over here to a different part of chromosome 12, which is over a whole lot of repetitive elements, when you look at the H3K9 binding after stress compared to control, it's really a very large increase. More raw data, again on chromosome 12 in a different region. If you look here, for example, over this bunch of repeats, and look at the comparison between the control animal and the stressed animal, the H3K9 binding, trimethyl binding, is obviously a lot greater in the stressed animal, and you have another example over here. So what Richard did then was to say, among all of the repetitive elements that he could identify, did they go up? Did the H3K9 enrichment go up in the stressed animal, or did they go down? And by an answer of something like 60,000 to just a few, the stressed animals had more. And then he identified some of the individual retrotransposons, like the lines, the signs, the long terminal repeat, and the other ones here, where he got really large increases in H3K9 enrichment in the stressed animal. And it may be as a result of that we got decreased synthesis in the hippocampus, decreased mRNA levels for certain retrotransposons. We haven't proven that the previous slide is the cause of this slide, but we would like to think that it is. And so here in the hippocampus, you have decreased expression of two retrotransposons that we measured at the messenger RNA level. We did not see that in the cortex or the cerebellum or muscle or heart or liver. So in conclusion, in hippocampal neurons, stress induces a broad increase in repressive marking of repeats and transposable elements. And acute stress promotes a strong and selective repression of these particular retrotransposons. And what Richard would like to do would be to weave our data and subsequent data that we can get into the concept of a genomic stress response, which would suggest that in the presence of environmental stress, there are maneuvers to promote genomic and transcriptomic stability. That's the argument. So this work was all collaborative between my lab and that of Bruce McEwen. And we depended absolutely on the level of knowledge and expertise and generosity of the Alice lab, and in particular, Tom Milne. Tom Milne is now at University of Cambridge, I think, or Oxford. And his ability to keep us going through this series of studies was a sine qua non of our getting the work done. I thank you very much.
We have plenty of time for questions if you've